Hi everyone, my name is Jian Zhou. I'm glad to share with you our recent work on sequence-based modeling of multiscale 3D genome architecture. A fascinating aspect about how our genome operates is that it folds hierarchically into highly ordered 3D genome structure at different spatial scales. And such multiscale architecture is well measured experimentally by Hi-C type sequencing-based techniques. If we visualize the Hi-C data of pairwise interactions between all pairs of genomic locations within genomic intervals of different sizes, we see those patterns as visualized in the center plots, each showing interaction patterns at a different spatial scale. At the more local scale, we observe individual chromatin loops, which are contacts between pairs of multiple genomic locations, and topologically associating domains, or TADs, where all positions within the same TAD tend to preferentially interact with each other. And at a larger scale, typically beyond 1 megabase pair, the most prominent interaction patterns are results of the chromatin compartments. The genome can be divided into compartment A and compartment B, and sequences within the same compartment preferentially interact with each other. I label these two compartments with different colors in the plot so you can also clearly see their preferential interaction patterns. This multi-scale genome architecture is associated with many aspects of the genome function. For example, the compartment A is highly enriched in transcriptionally active regions, and compartment B is typically enriched of transcriptionally inactive regions. And like all genomic location-specific phenomena, the multi-scale genome structure is almost surely highly dependent on the genomic sequence. Therefore, we are interested in developing machine learning models to predict the multi-scale genome structure directly from the sequence. Previously, two brilliant studies, DeepSea and Akita, have tackled the problem of modeling sub-megabase scale interaction patterns from the sequence, but it still remains a challenge to model the genome's architecture at all scales, including large structures such as the chromatin compartments, and utilize longer genomic sequences up to whole chromosomes. So before I show you our approach to tackle this challenge, I would like to first give you an example of the actual predictions from the model. Here we show a series of predictions made by the model at 6 different scales. Each heat map here shows a matrix of genome interactions from all pairs of genomic beans within a genomic interval with lengths from 1 megabase pair to 32 megabase pairs, as indicated on each plot. The genome interactions are represented by log fold over background, where the background is computed as the expected normalized contact scores at each genomic distance. And you can think of this series of predictions as zooming into the 1 megabase pair region from the 32 megabase pair region. And each plot shows the predictions zooming into the center half of the genomic region shown in the plot on its right side. At all scales, the size of those prediction matrices are fixed at 250 by 250. So for the more local predictions, they are predicted with higher resolution. The maximum resolution is 4 KB. And here we show the experimental measurements made by micro-C, a high-resolution Hi-C type technique. And we can see that they're pretty consistent with the predictions. All those predictions are made on test chromosomes that the model has never seen during training. These multi-scale predictions now allow us to predict genome structure at varying sizes, from the more local chromatin loops and TADs to chromatin compartments, which can only be seen at larger spatial scales. We also quantitatively measure the performance across 2 entire test chromosomes, with a correlation consistently ranging from 0.7 to 0.8 across all spatial scales. And you may have noticed here that we actually have predictions that are even larger than 32 megabase pairs. And indeed, our model can predict up to 256 megabase pairs, which is larger than the longest human chromosome. And in addition, we can also predict interchromosomal interactions at those scales while receiving multi-chromosomal input. So therefore, you can in principle predict almost all interactions that can be measured through Hi-C type techniques. So now you have seen a key idea behind our multi-scale sequence model, which is to model the genome interactions at multiple scales and multiple resolutions. This is important for scalability because it allows us to make fixed size predictions at all scales. And to enable those multi-scale predictions, we also made the model generate a series of sequence representations at the corresponding resolutions, starting from the raw sequence. Genome interaction predictions at each scale receives input from both the sequence representations at the same resolution and the predictions from the upper level, which introduce information from larger spatial context. The information flow in this model goes from the bottom-up order for generating sequence representations starting from the raw sequence, and it follows a top-down order for generating predictions starting from the largest spatial scale. The entire model is built with custom convolutional network architectures. This multi-scale, multi-resolution design also fits the nature of high-C type data because longer distance interactions can only be measured with lower spatial resolution because of the low number of reads. And this multi-resolution predictions also resembles how high-C data is typically stored in multi-resolution matrices. These models are trained to predict genome interactions at multiple scales and resolutions as measured by micro-C data from the genomic sequences. Going back to the model predictions, it's worth mentioning that the model can also predict different types of genome interactions. For example, here we show predictions for polycomb-mediated context on a test chromosome, and the contexts are indicated by the black arrows. Notably, these interactions are often cell type specific, and in this example, these interactions are predicted for H1ESC cell, but not for the HFF skin fibroblast cell, which are also consistent with the micro-C data for both cell types. And here is a different example where we show the enhancer promoter interactions that are predicted specifically for HFF cells, but not for H1ESC cells. Okay, so far we have focused on demonstrating the model predictions on sequences from the whole chromosomes. And this ability to predict from new sequences is really what enables the various applications of these models. One such example is predicting structural variant impacts on 3D genome interactions. Experimental studies of many pathogenic structural variants have shown that they can lead to genome structural changes that are relevant to disease phenotypes. And now with our model, we can in principle predict the effect of any structural variant at any size by computationally generating genome sequences that contain the variant and predict their 3D genome interactions. And here I will show you one such example where several structural variants in the same region cause several distinct limb malformation phenotypes. And all of those variants are larger than 0.9 megabase pair in length. Here we show the predictions for the genome interaction effects predicted for those structural variants. The wild-type genome are shown on the left, and the mutated genome after introducing the structural variants are shown on the right. And the structural variant positions for those deletion, inversion, and duplication variants are indicated by the black and gray bars on the left side of each of those plots. Interestingly, in all of those examples, the structural variant cause ectopie interaction from the same enhancer region as indicated by the blue arrows with different gene promoters, which are indicated by the black arrows. Indeed, this is consistent with the 4C6 experimental results, which showed gain of de novo interactions with the same regions that the model has predicted. As this example demonstrates, such techniques can be applied to various structural variants that we have no experimental data for to help us analyze their effects. Another important direction of application is applying the model to probe the sequence basis of 3D genome. Because the model has likely captured useful knowledge about the sequence dependencies of the 3D genome, it can be used to generate hypotheses about sequence-based mechanisms of the 3D genome by designing and performing what we call virtual genetic screens. Like a real genomic screen, we can design the library of sequence alterations that we want to introduce. generating the sequences and then obtain the predicted genome structure of those sequences. Because this is done computationally, we can do it in very high throughput, and we have almost no restrictions on the type of sequence changes that we want to introduce, making it highly flexible in the design. Specifically, we designed two categories of screens to probe the sequence dependencies at sub-megabase scale and chromatin compartment scale, respectively. And because of time, I will focus on compartment scale screen today. Indeed, one of the questions that I'm most curious about when developing those models is what are the sequence dependencies of chromatin compartments. To study this question with sequence models, we first trained the Orca model on an acute cohesin depleted Hi-C dataset. Acute cohesin depletion cleanly eliminates the TAT in this dataset, while the chromatin compartment remains largely unaffected or strengthened. This has allowed us to deconvolve the sequence impact on chromatin compartments from their impact on TAT. To identify sequences with activities on determining chromatin compartments, we designed an insertion screen approach by swapping in source genomic sequences into different target locations of the genome and measure their predicted impact within a 32 megabase pair window with structural impact scores. For each target location, we predicted the impact of inserting every one of the source sequences. Because the source sequences are generated by tiling the genome, we can plot the source sequence impact on chromatin compartment level genome interactions, like those plots shown in the bottom, where the x-axis indicate the genome coordinate of the source sequence original location. In this demonstration, we use source sequences tiling a 32 megabase per region as shown in the top right plot, and the 9 target locations are also from the same region as indicated by the arrows on the bottom. We observed that there are clearly two groups of target locations that are sensitive to different source sequences. It turned out that the red target locations are from the B compartment and are sensitive to B to A transition, while the blue target locations are from the A compartment and are sensitive to A to B transition. And the first observation is that most of the sequences that have B to A transition activities are from the A compartment, and A to B transition activities are widespread, but are more enriched in B compartment. To further study the properties of those sequence activities on chromatin compartments, we also perform additional screens. We first tested the minimum length of the source sequences that are necessary for possessing meaningful compartment alteration activities. And it turned out that the A to B transition requires relatively large sequences, at least 6 to 12 kilobase pair, while transition from B to A can happen with as small as 800 or even 400 sequences insertion. Importantly, analysis of those locations that have strong compartment A activity shows that those regions mostly coincide with active transcription start site with an odds ratio enrichment of more than 100-fold. And on the other hand, we also studied the length scale of sequence patterns required for possessing chromatin compartment activity by using a modified screen approach. In this screen, we permuted the source sequences by blocks of sequence segments of a specified size before inserting them into the target locations. Because all sequence patterns that are larger than the segment size are destroyed by the permutation, if a sequence still possesses activity after permuting k base pair segments, the sequence patterns that this activity depends upon is likely smaller than k base pair. We observe distinct sensitivities to sequence permutations for A and B compartment activity. Sequence with compartment A to B activities on the left is almost unaffected by permutations even up to 2 base pair segment size, indicating little dependency on complex sequence patterns. And consistent with this observation, the compartment B activity is correlated with single nucleotide composition with AT-rich regions possessing stronger compartment B activity. And in contrast, the compartment A activity is reduced by permutations around 100 to 200 base pairs and almost eliminated with smaller segment size, indicating that these sequence patterns around 100 to 200 base pairs are required for compartment A activity. Taken together, we propose the following model of the sequence spaces of chromatin compartment based on our sequence model, which can be further tested in experimental studies. In this model, compartment A formation is driven mainly by sequences at TSS, which likely introduce widespread chromatin changes and compartment A formation. In contrast, compartment B overall does not depend on complex sequence patterns beyond 80 content and requires extended stretch of sequences at least 6 to 12 kilobase pairs without compartment A activity. Finally, the sequence models of multiscale 3D genome structure can be applied to study various problems related to the 3D genome, including applications that we have explored here, such as identifying effects of structural variants on multiscale genome structure and perform virtual genetic screens. And it can also be potentially applied to directions that we have not explored yet, such as designing artificial sequences with desired genome structural properties. And you may be able to come up with creative applications that we have not thought about. Therefore, we have provided a web server and made the full code and models available, so you can use those to generate predictions from our multiscale 3D genome sequence models or train new models on your dataset of interest. I would like to acknowledge my funding sources and my lab members. Thank you for listening, and I'm happy to take any questions.